Like you've heard, SENS Foundation, located in Mountain View, California. Some people think of that as most well known for Google, but someday SENS is going to be the primary reason people think about Mountain View. You all are probably swimming in mitochondrial background and ideas, so I won't really tell you that much about it, except I just want to point out something that Richard touched on, which is that there's tons of copies of your mitochondrial genome in every cell. It really varies depending on the cell type and what that cell is up to and what it's being fed. But there's something like 1,000 mitochondria per cell, and somewhere between 2 and 10 mitochondrial genomes in every mitochondria. So you think of your genetic copy number as being 2, but your mitochondrial genetic copy number is something like 5,000. And keep that in mind. That'll come into play later. And you all already know about mitochondria producing energy and that leading to free radicals, which damage your mitochondria. And specifically, what I'm concerned about here is mitochondrial DNA mutations, much as in the last 2 talks. I just want to remind you something you were just hearing about was the 2 different ways that we make energy: glycolysis and oxidative phosphorylation, or anaerobic and aerobic respiration. The only thing I want to point out is that you make a lot more energy this way. It's more efficient, but your cells can do both. And I just want to mention one piece of trivia is that you can shove it in this direction by feeding cells galactose and force them to do glycolysis, to do oxidative phosphorylation. So keep that in mind. So you heard that 13 proteins are encoded by the mitochondrial genome in there, but mitochondria are huge organelles made up of many proteins, and so something like 99% of your proteins that make up your mitochondria are encoded in your nucleus, and those get targeted to the mitochondria using a mitochondrial targeting sequence or a mitochondrial localization sequence, same thing, and that's how they get there. Let me say something about these 13 genes. Certainly not coincidentally, evolution has kept these 13 there, and all 13 of these are involved in the oxidative phosphorylation pathway. We are working on 5 of those genes, ND1, ND4, cytochrome B, and ATP6 and 8. And like you heard about already, mutations in any of these genes, they're all essential genes, can have very severe phenotype, blindness diseases in complex 1. You heard about implication of Parkinson's disease with mitochondrial mutations. And you've also been hearing about the phenotypes primarily of mutations tend to be in the skeletal musculature or in the neuronal cells. And so these diseases of those organ systems. So what we want to do is our goal is to try to figure out a way to take these 13 genes and keep them safer. So take them out, not take them out, but make backup copies of them in the nuclear genome so that they're not exposed to all this horrible damage, oxidative stress in the mitochondria, and you put them in a nice safe form nucleus with DNA repair machinery and stuff like that. So we're taking 2 approaches to this. The first I'll have much more to talk about with. Second, basically just telling you that we're working on it. You heard about this 2 years ago from Michael Teitel at this conference. So I already told you, it's an easy problem, right? You just got to take these 13 and stick them in here and stick on an MTS, right? And everything's fine. Well, people thought of this 25 years ago, and it doesn't really work. There's been some incremental success with it, but people have had a difficult time getting effective protein import and rescue just by sticking an MTS on any of these 13 genes. And in fact, they sometimes result in toxicity, clogging up of the import pore machinery. So probably due to their biochemical hydrophobicity. So going back to the biology of those other 1,000 genes, it turns out that about half of those genes don't just have an MTS on them. They also have their transcripts, their mRNA, targeted near the surface of the mitochondria before they get translated. So once again, about half of your mitochondrial genes, their transcripts, localize near or at the surface of the mitochondria where they're translated by the cytosolic ribosomes, not the mitochondrial ribosomes, right? Mitochondria has its whole own protein synthesis machinery. You're still using the normal cytosolic machinery, but doing it right at the surface, maybe co-translationally. And so we want to take advantage of that. And so it's been somewhat worked out that it is the 5' and 3' ends of these transcripts that are targeted that are required for targeting. And so what we're doing is cutting pieces of taking an MTS and sticking it on the front end of the gene, taking the 3' untranslated region of mitochondrially localizing RNAs, sticking them on our nuclear recoded mitochondrial genes, targeting them to the mitochondria where the proteins will hopefully have an easier time being imported. This, we didn't make up this idea. Marisol Kroll-Dobrinsky was sort of the pioneer here, and partially under SENS Foundation support a number of years ago, she's published several papers demonstrating that to some extent it works. It's still very controversial, however. There's a lot of questions that remain unanswered about how it's working and how well it's working biochemically, how efficiently it's working, and even whether it's working at all. So we really wanted to double down on this to get much more data about how it's working, to try it out on different genes that she hasn't tried yet, and try and improve the efficiency even more. So this is our sort of plan of how we cloned these things. We took the mitochondrial genes. Like I said, you have to recode them for nuclear, the nuclear codon usage. And then we stuck an MTS on the front. We're using the COX-10 MTS and COX-10 3' UTR. And then as negative controls, we made all the possible combinations of either having or not having a MTS or the 3' UTR. And like I said, we were working on these 5 genes, and so we've made 20 different constructs just sort of as a basic, and put it in different expression vectors. And we've made cell lines for all of these genes, stable cell lines in 293 cells and force-nine fibroblasts, as well as in some patient cell lines, and I'll talk about a little bit about some of those in a moment. And of course, we confirmed that the DNAs were being stably integrated into the genome, and then we also confirmed that they're being expressed at the RNA level. So you recall that the whole trick that we're trying to do here relies on the idea that we can target the RNA to the surface of the mitochondria. So are we actually doing that? Well, we do mitochondrial purifications, but gentle enough purification that the ribosomes can still be associated with it. And then we do reverse transcription, PCR, and measure how much is actually there. And we get a very nice enrichment of our targeted construct, in this case cytochrome B, versus the untargeted control. And if you went by Ali's poster, you already know all about this. What about the protein? Protein, we have a little bit harder time getting a lot of it in there and detecting it directly, but we can detect it associating with the complexes by a co-immunoprecipitation. So if we IP down complex 5 with an antibody against a random member of complex 5, and then co-IP with an antibody against the tag that we tag this with, we can detect HV6 in complex 5 where it should be, and ND1 in complex 1 where it should be the top band there, not the bottom band. I told you before that there can be some toxicity from this, and we're trying to figure out the best ways to measure that. The cells don't all drop dead, that's good. But we want to look more directly at how the mitochondria is behaving. So we looked at the assembly of the complexes, and so this is a blue native gel, and you can see the complexes. I don't know how well you can see it. It's kind of blue on blue, but that's the nature of the experiment. The complexes are intact. They don't fall apart. And then we can do in-gel activity assays, so actually look at the enzymatic activity of the assay itself. And that's working just fine here in complex 5. We've done that for complex 1 and a little bit with complex 3 also. So then we wanted to see if we could actually do something useful with it. And so this is just one experiment that we did with a patient cell line. So you heard before about hybrids that people make from patients who have mutations in these in some of these genes. And so we got our hands on an ATP6 mutant cell line, and took our made stable cell lines expressing our positive and negative controls. But when we started playing with them in the lab, we couldn't really tell the difference between any of them. But we actually had a hard time telling the difference between the patient cell lines and wild type cell lines. For most of the assays that we can do in the lab, like these assays and like growth assays, and even growth on galactose, which, like I told you before, cells should only be able to do oxidative phosphorylation on it, but these cells could grow just fine. But we finally went and borrowed Gino Cortopacci's seahorse machine and measured their oxygen consumption. And you can see the wild type cells here doing great oxygen consumption. The mutant cells, okay, now we can finally see why they're sick, because they can do so much less oxphos than the wild type cells do. But unfortunately, we didn't get any rescue from our rescue construct. So we stopped and we thought about this, and this is actually a pretty mild mutation in ATP6. It's just a point mutation. The protein exists. It gets to the complex. We may be competing with the endogenous one, and the endogenous one may be partially functional. So we wanted to work with a cleaner system, and so we went looking in the literature for null mutations, which are very rare. But we found a couple that were reported, and we requested those cell lines, and there were mutations in cytochrome B and ATP8. And so we're really excited when we saw this, and we could take those mutant cytochrome B mutant cells and do a native Western and be able to detect complex 3 robustly on a Western. But then in the mutant cells, the complex 3 is totally gone. It's not just like band shifted or something. Without cytochrome B, the complex vanishes. These cells will just die out on galactose, which is great. And then similar with ATP8, we haven't done the native gels with it yet, but it seems to be a total null for the protein. We have an antibody against endogenous ATP8. So we started doing experiments like this, putting our constructs into the cytochrome B null cells and the ATP8 null cells. And we start getting partial results like this back. I'm not going to show you the data because, well, it may have been a false positive. We can't really tell the difference because when we went back to double check, we got really excited. But then when we did a PCR that can tell the difference between mutant and wild type versions of the cytochrome B gene, it turned out that this cell line was reverting back to wild type. And at this point in time, it was essentially half reverted. So that was really frustrating. So this slide's kind of a long story, but basically we went through the long, somewhat difficult job of making the cells truly homoplasmic. They were, at least the cytochrome B ones were like over 99% homoplasmic when we started, but then they can come all the way back to being wild type. So we did we built some pretty sensitive qPCR assays to be able to tell the difference between the wild type and the mutant cells, and then we used a mutagen to ethidium bromide to deplete mitochondrial DNA, and then you pick clones and select for screen for cells that have less wild type. And long story short, we found some, and then we got some educated guesses, and then we tested some of them, and we could get at least one that would completely die on galactose. So that's basically where we're at on that, is that we think we have some great tools that we can use to test these for rescue. So to summarize, we've made constructs for all 5 of these genes. We can make stable cell lines both in wild type and mutant cells. We can express them. We can get good RNA targeting and at least some targeting to the place where they're supposed to be, the right complex, the right oxphos complex. Another thing from Allie, the super intern this summer, that you may have seen on her poster, we wanted to confirm, are we using the best targeting signals? So we've been using COX-10 basically because Marisol was using it, and then there's some other ones that people have used in the literature. We want to compare them to those. Similarly, we found that COX-10 worked best, but then for kind of a long story, we wanted to try the ATP5G genes, and I'll leave it to Allie to tell you the whole story about why we suspected that one of these might work, and in fact, one of them seems to be working better than COX-10, so we might have a better construct to work with. Since I have a couple minutes left, I'll tell you about, we don't have any data, but that there's, like you just heard, that there's other possible ways to skin a cat, and there's yet another one that you haven't yet heard about at this conference, but you heard about it 2 years ago, and just basically letting you know that we're going to try playing around with the RNA import also. So what Dr. Teitel told you about 2 years ago was that you can, if you put a certain tag, a pretty simple tag on the front of an RNA, you can target it to the mitochondria, and not to the surface of the mitochondria like I was doing before, but it actually signals for import, and he laid out some of the mechanism by which it gets transferred from the outer membrane to the inner membrane, the matrix, and how it gets processed after that. But basically, we want to take advantage of that and try to use it on these 2 genes that we have great tools to study now. So we have antibodies, and we have cell lines that are true nulls for these genes. And so we've made these constructs with this simple stem loop on them and stuck them onto these 2 genes, and we're testing them out as we speak. And then also, since we're so good at targeting RNA to mitochondria already, we figure we should play around with these 3' UTRs and see if you can combine forces and maybe get even better efficiency. But I don't have any data for you on that yet. So to looking into the future, what we want to do is rescue some of these patient cell lines, and of course start looking, expressing more than one of these genes at the same time. See if you can rescue entire complexes, and go into mice, and then of course people, and there's, like you've heard about, there's the somatic accumulation of mitochondrial DNA damage, and also these really horrible debilitating childhood diseases that they're really waiting for treatment soon. So this is the wonderful team of people who does all the work, and some lab members past and present, and collaborators, and People have shared reagents and mentoring and advice. And everyone who volunteers and supports SENS Foundation, thank you so very much. And I'll stop there and take some questions.